I welcome those who have just joined. We may begin in more 5 to 10 minutes, waiting for others to join as well. Thank you. So while we wait for others to join, it would be really great if you all could tell us about your background, from what all fields are you coming, and also about your research interest. So I see new people joining us today. Hi Nadia, hi Pooja. Zahara, you are from one of our programs, right? For the Chem Informatics one. Hi.
Yes, I am in that program.
Hi, Zahara. It's good to have you here with us today.
Thank you so much.
Hi, Pankaj. It would be great if you tell me about your background and your research interest in the chat box so that we know we have people from which all fields. Thank you. We are live on YouTube and I've shared the link. So if any one of you misses out on the Zoom meeting, you all can watch the meeting over YouTube as well. I'll share the link again for those who have just joined. Here is the link. Thank you.
So, Nalika, it looks like you're on mute.
Oh, I'm so sorry. Yes. So I was saying that we would be starting in more 5 minutes. Thank you for joining. Hi, Dr. Vivek. It's good to have you with us today. We would want to know about your research interest and also your background. You come from which field? It would be great if you let us know in the chat box. Thank you. Okay, we have someone by the name of King. Hi, King. Hi, Hema. Hi, King. It's good to have you with us. Okay, so you are a public health microbiologist. That's interesting. Thanks for sharing. Thank you everyone for joining. I think we should begin now while others can join midway. So welcoming you all to today's webinar on COVID-19 genomics and variant program. We would, okay, I'm so sorry. So in this program, we will learn about the ways the SARS-CoV-2 pandemic has transformed our appreciation of genomics and bioinformatics, and understand how genomic data analysis tools can help identify specific viral strains and understand multiple sequence alignment, phylogenetic analysis, and the significance of mutations in the context of protein structure and function. My name is Sonalika Ray, and I'm the Omics Logic Community Manager, and I'm responsible for daily interaction with thousands of users that are a part of a bioinformatics community, which is managed by Pine Biotech. So before we start with the talk, I would want to introduce us. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. Omics Logic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. And I'm honored to have been working with a team that has collectively an amazing experience in this area, including the other co-founders of the company, Dr. Alfred Tauber, who comes from a medical background with an expertise in oncology, immunology, and biochemistry of inflammation. And one of the major projects of his is the establishment of Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonid Brodsky is the director of Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. Our platform is a result of hard work by some of the people mentioned here on the right. Julia Panov, who is an expert bioinformatician and has helped prepare many of the materials that we would use. Avi Titovsky, who is a computer scientist and also is an expert who developed the T-Bioinfo platform that we would be using. Dr. Harpreet has an expertise in the application of statistical machine learning techniques on biomedical data and the development of prediction tools and databases, and she looks after the curriculum development, bioinformatics, and data science course development. Dr. Mohit Mazumdar is an experienced bioinformatician with multiple publications in peer-reviewed journals and collaborations in industry and academia. And over the past several years, he has been developing collaborations with universities as a mentor and project lead for omics logic training and research fellowship. Now, I would like to pass it on to CEO Pine Biotech, Ilya Brodsky, to take the session ahead. Over to you, Ilya.
Thank you very much, Analika, and welcome everyone to the program. I'm going to share my screen and briefly introduce the program itself. So as you know, the surveillance of SARS-CoV-2 and specifically variants of concern is an ongoing effort that is critical to the successful strategy that will help overcome challenges related to vaccination, related to other factors that contribute to the emergence of novel variants, and to the ways that these could be addressed with prevention, treatment, vaccination, and other types of measures. So what we wanted to do in this program is to introduce all of the participants to data repositories, how to effectively use them, including repositories like NCBI virus and GISAID, where millions of sequences from around the world have been shared just in the context of this pandemic. But importantly, the rate of submission is continuing to grow. And we'll talk a lot more about what kind of data and how to find the right kind of data, relevant kind of data to structure a bioinformatics project. But we also will talk about some of the analysis tools that help us translate this data to information that is relevant. So what kind of methods are we going to be talking about? We're going to be talking about sequence alignment, gene expression studies, how to analyze the immune responses to the infection, how to organize our data in a phylogenetic tree and use molecular clock to kind of understand the timing that helps us establish some difference between sequences or groups of sequences. We'll talk about structural biology, how we can map these variants onto the structure of the critical proteins like the spike protein. And we'll talk about variant calling, how in all of this data that comes from different sources with different methods of sequencing, different depth of sequencing, how can we extract confidently the specific variants that are of interest? And then we'll also talk about how to utilize these methods using the T-Bioinfo platform, which allows for a seamless end-to-end processing, visualization, and analysis of sequencing data, but also how to use R to then prepare reporting on the identified variation and how to look at those reports following some examples of industry standards that are being used by teams around the world that are continuously engaged in this effort of monitoring the emerging variants and the development of variation in this virus. So here you can see on NCBI how NCBI right before the pandemic or maybe even during the pandemic has updated this interface for NCBI virus, makes it very easy to find specific sequences. So we'll actually go and do some hands-on exercises to find sequences from different animals, from different time points, compare different SARS-CoV-1 and 2 variants, or sorry, genomes, and how to then track the progression in time. So it's a very useful interface. And then down here below, you can also see some recent statistics on the number of sequences, genomic sequences that have been submitted for the novel coronavirus. So really a huge repository that all of us have access to. And we'll talk about how to gain access, how to know how to filter and find specific types of data that you need, and then how to also structure your bioinformatics research question using this information, and perhaps talk to you about the questions you might already have and think through how can we organize that and translate that into a technical sequence of steps that you can follow to actually get reliable and meaningful results from this information. So the important outcomes for this program is to study trends behind the emergence of SARS-CoV-2 variants. We'll talk about the emergence of the virus, the infection itself, where did it come from, and how do we know where it could have come from. We're not going to be addressing any kind of speculation about where we can know the specific location of the emergence or the time of the emergence, but we'll talk about how do we use genomic data to know the source, to identify a common ancestor. We'll talk about genomic surveillance. What does genomic surveillance mean? How is data collected? Different types of sequencing technologies, different types of bioinformatics analysis methods. We'll talk about downstream analysis of genomic data, so how we can use information from genomic data sets to understand phylogeny or evolution. How do we track origin, and how do we report accurately the type of analysis that we've done? We'll talk about creating a report on genomic sequencing data using cloud processing and a custom R script. So I'll talk a little bit more about that in a few minutes. And we'll talk about future directions for these large-scale vaccination efforts and variant of concern monitoring. So where is it going, and what are some of the factors that we should be mindful of as we track the emergence of existing and novel variants of concern, and some ideas about classification and why it's an important effort. So to start it off, I want to briefly kind of go back to where we came from. How did we actually get to this point of knowing that there are many different variants and many different variants of concern, and what's the difference? So as you can see here, probably most of you are already familiar with this type of visualization. This is a phylogenetic tree that shows us how over time on the x-axis we see emergence of different groups that right here are already colored by their phylogenetic taxonomy. So these are the different names of strains. And you can see down at the bottom the proportion of these strains in the worldwide population. So we originally have been introduced already to this idea that genomic data can provide us with a time series type of analysis that demonstrates how variants of concern have changed in proportion of infected cases. Now, there are many issues around this specific type of reporting from a data perspective. Of course, there's the sampling rate, there are the specific methods that have been used to determine association with different taxonomic units. And so that is why essentially it is very critical to look at this data and look at this type of reporting with in-depth understanding. So for example, here you can see how the number of types of clades that have been reported is growing over the course of this pandemic, from 3, then to 5, to then 8, and then to 10. And so that number will continue to grow, and the specific proportions here are important as well. So you can see that some are completely going to disappear over time, some are going to emerge and become completely new. And so over time, the dynamic of emergence and reappearance of some of these variants is called phylodynamics. So this is a phylodynamic view of this SARS-CoV-2 phylogeny. So we conduct a phylogenetic analysis, we look at genomic variation, and then we include the component of time. So really the objective of this analysis is to try to identify the factors that drive this variation as it emerges across time. And so because of the availability of data and because of this universal approach to taxonomic classification, we now can look specifically at each type of variant to understand how they individually develop over time. So what you can see here are the specific variants that have a proportion by country. And you can see that most of the countries are going up, but not in a linear and not in a uniform fashion. So you can see that there's a lot of variation by region and country. You can also see the specific mutations that define a particular clade or a strain. So here you can see specific variants of concern down at the bottom, the famous ones like the UK, South Africa, and Brazil. And you can also see how these mutations are mapped onto 3D structures. So all of this information is widely accessible. And I think what is the major point is for us to be able to track the data as it emerges and to be able to apply these meaningful analytical techniques to gain a better understanding of the biology behind the individual variants, to understand the complexity of this taxonomic classification, and then to be able to sample new data that is coming out and find relevant information in this new data that we can make decisions based on. So what's important is obviously to also think about these new developments. And all of us have been hearing not so long ago, this novel Delta variant emerged in in India, not so far ago, and not so long ago. And this new Delta variant, the B1617.2, how it has been growing tremendously. I think by June has accounted for about 70% of all the new infections. So it's a major concern, especially because the vaccine effectiveness against this new Delta variant seems to be incomplete. So it does not offer 100% or even a 95% as originally was tested for Pfizer. For AstraZeneca, even lower, 60% effective. Remember that FDA approval is after typically 70%. So really major challenges to these standard approaches to one type of a vaccine, and needing the additional effort to really understand the phylodynamics. How is this strain going to continue to evolve? Are there new strains that are going to be added on to this B16172? So importantly, if we go back to this slide, you can see that major mutations, non-synonymous as well as synonymous, are not unique for each strain. In fact, all of these defining mutations overlap in many cases between the individual strains that are reported. For example, you can even see in the naming B117, B1351, right? So the B was one of the original clades that separated from the A clade. And so as we continue, accumulation of novel mutations means that this is not a completely new strain. It's really additional numbers of mutations that we need to understand their impact collectively on this effort to address the problem with vaccines. Not only that, important new changes to this new strain is that it affects much more significantly the younger population, which was not true for the original strain. It also tends to be a problem for people that have 1 or even 2 shots of the vaccine, and it is spreading much faster. So there are additional challenges that we should be anticipating as this process continues. But to really study this problem or this challenge in detail, I think we need to go back, understand the biology behind SARS-CoV-2, the infection itself, the rate of transmission, as well as the evolutionary changes that are starting to appear in the genome, and to understand this biology at the level of reporting so that we can accurately look and sample at data, and we can start asking questions about these individual mutations. So here what you can see is the organization of the genome. The genome is about 30,000 nucleotides long. It has a number of different proteins that are encoded in genes. So some genes are protein coding genes and some are non-protein coding genes. And what we want to do is find an approach that allows us to identify some significant mutations that you can see here higher on the y-axis, and start asking questions about the position on the genome, their rate of change, and how can we associate this with phenotype that ultimately could be defined in several ways. One way that we could characterize these mutations is to start looking at how these mutations are present on the 3D structure. And so by understanding the 3D structure and how it functions, how it works, we can start asking questions about whether we understand what the potential impact of this mutation is on the interaction between the viral and host protein interaction. Another way that we can study this is to look at experiments that show how genome replication rate changes because of these mutations. So here's an example from that kind of a study. You can essentially infect cells and track the number of genomes, and some interpretation about that rate and change is also important. We can also look at the type of change. So we can ask the question whether a specific nucleotide changing to a different nucleotide is also going to result in a change in the amino acid, and what kind of a change in the amino acid will it be? How evolutionarily likely is that change based on Blossom scores, or how the physicochemical properties of that specific change could affect the interaction again with the different proteins. So many different types of questions that we can ask, but ultimately we need data. And so to be able to ask these kinds of questions, I think we can rely, first of all, on scientific literature that has been peer-reviewed. And as you know, there have been thousands and thousands of publications that have been coming out at a very rapid speed to tell us about novel insights about the virus itself, individual proteins, as well as the interaction with the host. So what we want to focus on in this program is to take you through a number of curated data sets and associated publications that will help us set a context for the SARS-CoV-2 emerging variants of concern, as well as some of the aspects of vaccine design, drug design, immune responses, and other factors that we think are critical for our understanding of how this genomic data could be useful and how it could offer new insights and important insights on the ground about this effort that is ongoing. So one thing that we will talk about in detail is the organization of this spike protein, how the protein is organized, what do we know about the protein and its function, how can we ultimately look at the interaction between the viral proteins and the host proteins, and ask questions about specific regions or functional domains that are important in this process of interaction. In addition to that, we will then take a look at the connection between genomic data, as you can see here at the bottom, and the protein data that is available to start asking the question about rate of change or mutation frequency that is observed by sampling specific groups of genomic sequences available in the public domain. We will also talk about what are some of the potential selective pressures that could explain the emergence of specific mutations that we observe. So we will talk about how antiviral drugs have been applied, how host immune responses might be responsible for specific changes, and then try to link that with genomic information that we have about specific types of genomic genomes from the SARS-CoV-2 databases so that we can link those processes together. And ultimately, ask the question of specific mutation patterns that could explain what we observe in the drug treatment, in the vaccine treatment, in the host immunity patterns that are already being studied. And so this information is also important because many early stage studies about the immune responses that are typical, that are maybe abnormal in acute infection or typical for mild or asymptomatic infection can really start putting together the pieces of the puzzle for us to be more informed about how the interaction with the virus happens in the course of the disease, how it affects outcomes, and then start using that information to understand the specifics of these novel variants of concern. So what's good about this is that there are many publications already, and these publications come with a lot of data sets that we can all access. So ultimately, we will not only look at the reported clinical genomic information that is sampled from populations at different time points, but we will also take a look at the more research data that is available for cell line experiments animal experiments, as well as experiments done on human cell lines and from downstream patient studies. So we will talk about all of these aspects in the program and try to link the information that is being reported in the specific types of publications that also goes beyond these individual point mutations, but also talks about deletions and also multinucleotide changes that are observed in these sequences, which is an important part of information that is present in this genomic data, but is not as often being reported or spoken about as prominently as we hear about these point mutations that might have a consequence on the protein structure. So we will try to look at the clinical side and public health side, but also we will dive deeper into the research side and try to understand what other types of variations in data are present and how should we utilize this information in the context of the genomic data that everyone else is speaking about. So the program itself is a part of our standard Onyx Logic Bioinformatics training program called Bioinformatics for Infectious Diseases, but it has a much more specific and in-depth focus on the SARS-CoV-2 genomic data, transcriptomic data, structural data, and specifically with this additional focus on the variants of concern. And so we will introduce you to some of the important efforts that are being done throughout this country in the US as well as worldwide, where there are specific objectives to leverage some of the genomic surveillance for informed decisions that could be done by the US public and academic institutions to really address this challenge of novel variants. And to practice for the hands-on portion of the program, we will also talk about specific analytical tools that will include consensus level analysis of variants. This is something that you can use in your research work. This is something that you can use to design an independent bioinformatics project as a citizen science or as a scientist or as a student, and also something that you could leverage to gain skills in all of these aspects of analysis. So what are these specific types of activities that we will do? We will perform consensus level analysis of variants. So if you can imagine a sequencing data set with specific depth, how do you identify the consensus of where those variants are present in all of the reads or in the larger proportion of the reads? How to detect novel and prevalent variants, which means that we will look for specific positions that have been reported and find whether those are present or not and at which proportion in given samples. We will also look at subconsensus and low frequency variants, which is an important part that we already talked about, that ultimately the emergence of these prevalent variants starts with a presence of low frequency mutations that eventually gain prominence. And so that really demonstrates the evolutionary kind of struggle that happens between the host and the virus, where the progeny of the original viral strain divides up into these subclades, and then some clade becomes more efficient at overcoming the immune pressures, ultimately becoming the dominant strain. So we'll look at that subconsensus analysis and how to detect low frequency variants. We'll also talk about low quality samples. One of the major things that is ongoing right now is sampling data from wastewater and taking data sets that might not have high level of accuracy like Illumina sequencing, but instead Oxford nanopore sequencing. And how can we leverage that same data using some quality control measures to identify variants of concern? And then we'll also look at population scale, tracking the spread and emergence of variants in densely populated areas. So we'll take some curated project examples for that. Now, as we go through these topics, we will also touch on some of the basic concepts that you have to know to be able to participate in such studies. And that includes an understanding of different types of sequencing techniques, understanding of data types and data file formats. We will also talk about the specific details that allow us to leverage quality control in a meaningful way. We will talk about the process of sequence alignment based on different types of data, like FASTQ or FASTA files. We'll study specific types of protocols that will give us a better sense of how data is generated and what types of steps are needed to use that data in a meaningful way. And then we'll actually go through some examples of wastewater sequencing and other types of sequencing, and what are the sources and the outcomes for each type of study. And we will introduce you to some user-friendly tools that are available on the T-Bioinfo platform to perform multiple types of bioinformatic pipelines that cover the phylogenetic analysis, variant calling, gene expression, and structural biology aspects of bioinformatics workflow that are important to know in the context of such studies. So ultimately, what that means is building pipelines from logical steps, understanding how these logical steps tie together, and knowing what are the inputs and outputs so that your analysis is meaningful and reproducible. For example, one of the useful kind of views, I think, that has been on everyone's mind is, how do we identify the origins of SARS-CoV-2 in terms of zoonotic transmission? And so here we will turn to some of the published data sets and published papers that help us use the logic behind viral emergence and biology, and bioinformatics methods that allow us to study similarity between sequences to ask questions about the potential or the likelihood of zoonotic transmission between different known species. On the other hand, we will also look at some of the well-studied and known examples that show specific changes at the nucleotide level and the amino acid level to really understand how we can come to conclusions that have been reported in the public domain. And this mutation, the 614 gene mutation that was well studied and I think well understood, has really become a great example of how we can link the phylogenetic information, the specific variant calling information that we can focus on, and the biological understanding of its structural and physicochemical interaction properties that we can translate into a meaningful insight about an identified mutation. Now, this is a single mutation that we were talking about. But ultimately, when you look at data, you are present with a lot of information for each individual sample that contains many numbers of mutations that are typically present in a given sample. And so one of the challenges that you will come across is, how do I interpret this in a meaningful way, knowing that many of these mutations are either reported in literature and databases or not reported. They could lead to structural variation or not lead to structural variation. And then how do we look at them together, complementary to each other or detrimental to each other, to ultimately link it to a specific phenotypic change? And so that's why all of these steps, these analytical steps, are very important. So we'll take some time to study pre-processing and quality assurance aspects of sequencing data, and then we will turn to generating a report, a meaningful report based on what is standard or kind of something that people use in the industry and the research in general. So this is an example of a report from Cosmos ID, a commercial report that one might get from sending your data to a sequencing lab. And we will perform the exact same analysis using tools in R. So this is the report that we will generate using R scripts that take process data from a pipeline and turn it into a visual and statistically significant report that we can share with our collaborators and partners. And so to produce this report, we will actually go through the code and learn about some useful packages that are typically used for visualizing and performing statistical analysis of data, including ggplot2 and some other libraries for managing and working with your raw data, including looking at the specific positions that are associated with a variant, like the US or the UK, South Africa, and Brazil variants, including the Delta and the Delta Plus variants. And so this is going to be the culmination of this program, where you will be able to sample some raw sequencing data, load it into the pipeline, produce the output of the pipeline, and then analyze that whole pipeline output to generate a report that you can share in a PDF or a full report that you both understand the statistical prevalence of variants of concern, but also their biological relevance. Now, let's briefly go over some of the program resources so that everyone is clear on what is going to be included in this program. But before that, I would like to launch a poll and briefly ask you a few questions about your background and your interest in this program. So I have launched a poll, and I would appreciate if you could take a couple of minutes to answer some of these questions. So there's actually just two questions here. The first one is about your background or your designation. So are you a high school student, undergraduate student, a bachelor's degree in college, graduate student, a master's degree in college, postgraduate PhD, postdoc or research, faculty, or management and administration? And we use this information to continuously improve our programs and make sure that the level of complexity and technical information that we provide in the programs is appropriate for the types of people that are interested in these topics. So thank you for those of you that are answering that question. And then the second question is about your research interests. So Pine Biotech is operating a number of different programs through the omics logic training that we provide. And those include infectious diseases, data science training, machine learning, genomics, proteomics, epigenetics, transcriptomics, agribioinformatics, and general bioinformatics. So based on your responses here, we will be able to adapt our novel programs that are coming out very soon, and we will be happy to share with you some details about some of the upcoming programs that we have starting this week and in the next couple of months. So I'll just give it another couple of minutes for everyone to be able to finish the responses. Thank you again for taking the time. Okay, and let's take another minute to complete this poll. Please, for all of you that have not completed the poll, please take just a couple of minutes to finish your answers. Okay, thank you, everyone. It looks like we have 70% that have successfully completed the poll. So thank you, everyone, for sharing that information with us. I will end the poll for now. And also, before we actually continue with the program resources, I would like to pass it on to Dr. Harpreet Kaur, who's going to be one of the mentors in the program, to share a little bit of information about her background and how she will be able to help you work with your data and learn about these bioinformatics methods. Over to you, Dr. Harpreet.
Thank you, Ilya. Let me share my screen. Hello, everyone. My name is Harpreet Kaur, and I have a PhD in bioinformatics. My PhD research was on biomarker discovery for the diagnosis and the prognosis of the liver cancer using bioinformatics techniques. Now, let me give you some glimpse how bioinformatics can be helpful in identifying genomic signatures for identifying a specific signature for a disease condition. So by taking an example from my PhD research work, so for example, here in the first project, we use large-scale transcriptomics data to identify diagnostic biomarker panel for the hepatocellular carcinoma, which is primary liver malignancy. So here we employed nearly 30 datasets containing 4,000 samples to elucidate transcriptomic biomarker based on various machine learning techniques. In the second part, we focused on another primary liver malignancy, which is cholangiocarcinoma. So here we have identified 3 gene-based signature, which is LAMP-C2, ITZ, and C2, to discriminate cholangiocarcinoma for HCC in the normal samples. In the next part, we focused on another project where we want to classify and want to predict tumor at an early stage. So here we have identified 51 feature-based signature, which include 30 RNA transcript and 21 methylation CpG sites, which can predict hepatocellular carcinoma at an early stage with nearly 80% accuracy. And we have developed all the prediction model using the publicly available data and implementing various bioinformatics techniques. So in my current role, I'm curriculum developer and research consultant with Bioinformatics. I'm involved in designing research projects as well as assisting the participants in their analysis. And I also got the opportunity to co-mentor a number of the research fellow in their research projects, which are depending upon based on the publicly available data, implementing various bioinformatics techniques. So my current, apart from the projects on oncology, then we also we have developed various projects based on neuroscience. So apart from the graduate and PhD students, then there are some of the students who are actually high school students who completed research projects based on the publicly available data and using various bioinformatics tools that are integrated on T-Bioinfo server on the Bioinformatics. So my current research interest lies in the application of the bioinformatics in different human health aspects. Although my primary research interest in cancer genomics, besides oncology, infectious disease and neuroscience are the other current research areas where I'm working on currently. And I'm looking forward to being your mentor in the upcoming program. So now I would like to pass this session to Sanalika. So over to you, Sanalika.
Thank you so much, Dr. Harpreet, for telling us about your research area. Now I would like to welcome Dr. Mohit to take the session details ahead. Over to you, Dr. Mohit.
Actually, I think I will just do that part. I think Mohit did not.
Okay.
Okay, so thank you again. Dr. Harpreet is going to be our mentor that will help all of the participants perform hands-on analysis and also take on some of the sessions that we have planned. To introduce you to the specifics of the program, how you can know about the topics that we will cover, the schedule, how to register, all of that information, I will actually share that information with you. So if you could go with me to our site called learnomiclogic.com. And so for this part of the session, I would like to ask you to actually make sure that you are registered. So this is the link, learnomiclogic.com. So if you could please let me know once you have registered. On the site, I will show you how to access the specific tutorials and asynchronous materials that you will leverage throughout the program. I will also briefly demonstrate how you can access the T-Bioinfo platform and try the analysis even in today's session, so you get a sense of what kind of hands-on practical activities you will be going through. I will also show you how to utilize the R code playground so that you can practice some of the code that we will be going through for reporting and for tracking those variants. And also, I will then show you how you can register for the full program. So please let me know in the chat once you have been registered. If you're registered already, that's great. Just let me know in the chat. I will be looking for your answers. So once you are on this page, you need to go to login, and on the login screen, you can either log in using your email address or create an account right here. So I will log in using my details. And once you are logged in, you will be seeing your profile here where you can understand how the program actually works in both asynchronous mode and synchronous mode. So for the asynchronous mode, really you have access to a number of different opportunities for learning different courses. We have a number of different courses that use data from the public domain and show steps that you can replicate on your own. And another section that you will see here is programs. So the same is available on the top menu. You can go to courses and search for courses. So we can search here, for example, for SARS, and you will find this project right here, COVID-19 Origin and Pathogenesis of SARS-CoV-2. Or you can search for genomics and find a course that introduces you to the basics of genomics, how to align sequences, what are nucleotides, what are amino acids, what is multiple sequence alignment, what is variant calling, etc. You'll also find here some coursework on coding, including getting started with bioinformatics in R or in Python. Okay, so again, this is the main area for you to gain access to some asynchronous materials, and you can search here. Most of these courses are going to include some basic material that you can get started with. And then as you are convinced that this is the right topic for you, the next stage is for you to find some premium coursework that includes a little bit more hands-on and a little bit more depth on the various options for bioinformatics analysis, and that comes with our starter subscription. So the starter subscription is really affordable anywhere around the world. It's only $10 a month, and for that $10 a month, you gain access to all of the coursework that is available here, even though most of it you could sample completely free of charge. Then we recommend moving on to the next level, which are the programs. And so today we have been speaking about the SARS-CoV-2 program. So that program is going to start on July 12th. So if you search in the program, COVID-19 Genomics and Variant Analysis, this is the program that we're going to be starting. Now on my screen, you see what it looks like for someone that is already registered for the program. So let me introduce a little bit what this works like, or how this works like. So as you can see, we have listed here the different sessions. They start on July 12th, and you will see the topics listed right here with the specific dates for then when they will take place. And you can actually download all of this information by clicking on this icon right here. So you can scroll through and see how we will go through genomic data analysis. You will see how we will perform the analysis in R, and then you will see what are the different topics that will be covered in the different sessions. Down here below, you will also see some of the coursework that's going to be included, and that coursework starts with a course on Introduction to Bioinformatics. This is a free course that we recommend everyone to take, and the course includes some important information about the applications of bioinformatics in a variety of domains. So whether you are interested specifically in SARS-CoV-2, or you're interested in other topics like transcriptomic data analysis, metagenomic data analysis, precision medicine, and others, you can actually go through this course and see the different applications that will link it to the next step of what you can do in this process. So after you've sampled some of those courses, you will actually get a certificate. To get that certificate, you need to have complete all of the sessions that we have in the program. So there are going to be 10 sessions, and that will come with our point system that you need to, let me empty the caches here, that you need to complete in order to gain the certificate. So the certificate comes as a collaboration with a university in Haifa, which is a university that is focused on development of bioinformatics tools that it seems, Sonalika, that somebody forgot to upload the certificate here. But it's important to complete this information that is presented in the program to gain that certificate. And then finally, what you will find here is a Slack channel that you can join to be connected with the mentors and the community of those that are participating in the program. And so if you join the program by the start of the session, which is going to be July 12th, you will have full access to our mentors and to the other participants, and you will see all of the interaction happen in live sessions like today, as well as on a Slack channel project dedicated to the community. As you can see here, we have different Slack channels for different programs. And so there, it's an effective way to track different questions and technical issues, as well as to interact personally one-on-one with those that are members of this community. In addition to this, I want to show you a little bit about the Bioinformatics Toolkit that you will gain access to as a part of your registration. So registering for this program will give you access to some important pipelines that are going to be useful as we go through the different examples. So let me show you what I mean by that. Here, and by the way, after you have registered, you already have a demo account on this platform. This platform is called T-Bioinfo, and it is developed by the Tauber Bioinformatics Research Center. So here you can see how using different types of pre-made pipelines, you'll be able to understand better what are the specifics of the logic behind this analysis. So how does that work? You will gain access to this interface. In the interface, we have informative pop-ups that allow you to learn about each step of the analysis, including what type of data we are using, what is the logic behind each individual step, and what are the specific outcomes of this step in the whole process of analysis. So performing these pipelines together, you will learn about how the specific types of methods that we are utilizing are helping us gain a full result that we can analyze in the next step of the program. So once you have this result, some of this result is going to come as a PDF that you can download and explore visually. Some information is going to be FASTA files or even tables that you need to know how to work with. So to do that, I will give you just one example of what you can do with some of this information. And to do that, let me show you a course called Getting Started with Bioinformatics in R. So this is a course in R that will help you take FASTA files and tables and learn how to manipulate them to produce visual outputs. And the important part of this is that the practice comes right inside the browser. So even before you install and configure all of your libraries, you can already learn the code and try yourself. So for example, in this brief lesson, you will learn about different libraries to work with FASTA files and sequences, and you will see how the specific steps of this process are explained very clearly in terms of the coding. And then you'll be able to try it yourself, simply running this code in the browser itself and looking at the output. So learning about the code and then knowing how to perform the analysis itself gives you the ability to try this, and then you will be given some assignments that are going to be challenges for you to address as you try to write your own code. We'll help you reestablish what is the logic, but also how to practically translate that logic to coding skills in R. So the program itself will cover these topics from biology to bioinformatics to then the application to specific data sets that we've curated specifically for this program. And we'll end with your ability to perform the full analysis independently. And ultimately, for those that are interested, we also offer this as a transfer into our research fellowship program, which is a specific program that helps you analyze the data and then generate a report that you can share with others, including writing a full report as you go through the project steps. And to help you do that, we actually have a full project on how you can get started with a bioinformatics research project. So this project, this course, will guide you through the important steps of finding a relevant topic by performing literature review, identifying a data set in the public repository that you can analyze, and then submit the full project and get feedback from our expert community. So ultimately, when you go back to your personal profile, you will be able to track your progress and activity using our tracker and the point system. You will also be able to see what courses you have completed, and all the certificates for each individual course are going to be available on your profile as well, right here under certificates. You will also see the progress of the program and each session that you have completed, so you can also include certificates for the program itself. And finally, you will be able to follow our process to get a project completed during this program and post it under your profile. So once your profile is complete, and you have reached at least 10,000 points by completing the coursework assigned to you as a part of this program or independently, then you will be able to submit a full project, and you first have to learn about that project submission, and then submit your project as an example to all of the people that you would like to share it with that illustrates how you have performed this analysis. So here at the bottom of the homepage, you will find some of those examples from our previous participants that have completed a project. I think this gives a brief overview of what the program is going to be about, and hopefully everyone was able to see the different aspects of the program curriculum, the way that we will interact, and the different resources that are going to be included. If you have any questions, I would be happy to answer those at this time. And I do want to recommend for all of you that are interested in the SARS-CoV-2 project, we do have that project available that you can try out and see what that content looks like, learn about some of the basics as you get started, kind of setting the background for the whole project context, and then taking that forward with this guided, mentor-guided program if you're interested to learn more and also become proficient in some of these methods of analysis. How do you access the ARC course? So all you need to do is to register on the Omics Logic program platform, and you will see different courses as I've shown. So you can actually go to the courses tab right here and search for R. Search for R and getting started with bioinformatics. Or if you want more advanced R, you can go to Introduction to Data Science. Now, several of the projects that you see here also include specific codes in R. For example, if you go to Genomics, as I mentioned, you will find here some coursework in R as well. So whenever you see R code, that means that the course includes some practical coding in R.
Thank you so much, Ilya. So if you all have any doubts, you can ask now, or you can mail me at marketing@. I'm so sorry. Yes. Thank you so much, everyone. And thank you, Ilya, for guiding us through the session. Now, I would like to ask if any one of you have any doubts, you may ask now. But if you would want to address them later, you can mail me at marketing@pine.bio or mohit@pine.bio, and we would be there to guide you through your enrollment. Thank you so much, everyone, for being a part of the webinar today. And we're looking forward to your participation in the sessions and then learn together. Thank you.